Hello, my name is Eric Frink. I'm a fourth year biochemistry student at the University of Minnesota in the College of Biological Sciences, and this is my summer 2024 Europe project. So I did my Europe project on developing a live cell imaging method to quantify drug therapeutic targeting of non-proliferating persister cancer cells. A brief overview of this project is that cancer cells that have evaded drug therapy can persist in a cytostatic or non-prolific state. To assess the efficacy of a cell targeting approach under cytostatic conditions, it is necessary to have a method to accurately identify these non-prolific cells and their fate upon drug treatment. So what I did was I combined live cell imaging using a nucleus marker with two different software packages, Stardist and TrackMate, to optimize a methodology capable of identifying and tracing individual cell division over a time-lapse video for over 48 hours. I've also begun to apply this methodology to analyze the targeting efficacy of a particular histone deacetylase inhibitor in a persister cancer cell model of estrogen receptor positive breast cancer cells under endocrine therapy. Um, Eventually, this methodology will be applied to evaluate the specific targeting efficacy of therapeutic agents on non-prolific cell populations. So my materials and methods for this project, I used MCF7 cell line, which is a ER positive breast cancer cell line. These were marked with H2B GFP or a histone green fluorescent protein that is overexpressed in the nucleus of the cell, which allows for marking and imaging. And then for endocrine therapy, I used 4-hydroxytamoxifen, which is an active metabolite of tamoxifen, a standard endocrine therapy um, in practice in clinical use. And then I also used panobinostat, which is the histone deacetylase inhibitor that I mentioned. And then for my image analysis softwares, I used Fiji, which is an open source program that uses ImageJ, and then two software plugins for this open source software, Stardist and TrackMate. And then in order to develop this methodology, I cultivated cells in a DMM high glucose media and ensured they were healthy and proliferating properly. And then I used the Tekken Spark Cyto machine to image the same frame of cells within each specific well for 20 minutes every 60 hours, using both white light or like normal light, also called bright field, and green fluorescent channels. And then took these time lapse, or I guess simple images, and optimized parameters in Stardust to minimize the over and under counting of the cells. And then the TrackMate, I optimized the parameters to properly identify cell replications over the time lapse. Um, so this is an example of a bright field image or normal light image of cells, as well as an example of green fluorescence. Um, what the bright field shows is the entire cell. However, there can be an issue. If you notice in the circled in yellow, the cells can clump. But so then the green fluorescence nucleus marker helps to isolate some of that clumping, as well as identify just the cells when they're dividing. So, for example, in this for a nucleus, a splitting event recognition using the Stardust and the TrackMate, we can zoom in on this quadrant up here on these four cells, where one of them is undergoing a division event. And so in the first image, we have the base cell, and then second, it starts to condense. And then here it splits. And then the fourth one, the cells grow into two different cells healthily. And it recognizes the splitting event. And so for in application of this recognition process, I use the 4-hydroxytamoxifen and the panabiniser or the HDAC inhibitor on a MCF7 or ER positive breast cancer cell line. And so to mimic an endocrine therapy, which is used in clinic to treat breast cancer, I had to do a daily treatment of the cells with the tamoxifen uh, to mimic this. And then I had to, on day 7, so once the cells are properly arrested on day 7, the HDAC inhibitor can be added, or the vehicle, which in this case was DMSO. And then once 24 hours after this initial treatment is done, imaging can start. Now, oil does have to be added over the top of the media so that it does not dry out for the 72 hours. That's another difference, is that the cells were imaged for 72 hours rather than 60. And in this case, my method was to, instead of analyzing the entire time lapse of images, I just took 3 time points at 24 hours, 48, and 72 in order to count the average cell death at each time point. However, this method of counting underrepresents cells that do not show the propitium iodide. So propitium iodide is the marker for cell death. It can penetrate the nucleus when the cell is dying. But this method of counting, it does not represent the percentage of cells that do not express the propitium iodine fluorescence in between each time point. And so this method of doing so is sort of a preliminary analysis to the tracking methodology of this project, because, and sort of a continuation of this project, is that we use this methodology to track each cell and its fate, whether it dies or or if it divides, in order to capture the full picture. And then so the another future direction of this, more long term, is using this tracking methodology to assess the efficacy of specific targeting drug therapeutics on non-proliferative persister cancer cells, specifically either this HDAC inhibitor, but also on different ER positive cell lines, such as KAMA1 or T47D, as well as using alternative endocrine therapies such as fulvestrant.